Here I've got a nucleic acid strand, and let's say I want to synthesize another nucleic acid strand. There's three rules that you need to keep in mind when thinking about synthesizing another strand and creating a nucleic acid. Rule number one is you need a template strand. So this strand right here is going to be our template. Rule number two, the strand needs to be synthesized in the 5' to 3' direction. What that means is that we need to add nucleotides by using the hydroxyl functional group on the 3' carbon. Hopefully you'll be able to see that once I draw the synthesis out. And rule number three is you need to synthesize your nucleic acid strand so that it is anti-parallel to the template strand. And so what I mean by that is I could lay down another nucleotide across from my template strand this way or this way. And one way would be correct and the other way would be incorrect. Do you know which way would be correct? Well, if not, the first thing that you need to do, make sure that you can identify the orientation of the template strand. If you look up here, this is a hydroxyl that's coming off of carbon number 3 on the nucleotide. And I know it's carbon number 3 because carbon number 1 is always connected to the base. Carbon number 5 is always connected to the phosphate group. So this must be 4, 3, 2. On the other side is my carbon number 5 at the end of that nucleic acid strand. So what that means is that this end is the 3 end, the right end is the 5 end. So this orientation is incorrect. Hopefully it's obvious just by looking at it. The nucleotides are facing the same way. They are not anti-parallel, they are parallel. Anti-parallel means facing in the opposite direction. We also know that these are parallel and not anti-parallel because I have the OH sticking out of carbon number 3 at the end, making this the 3 end. You cannot have, for two strands that are base pairing, you cannot have the 3 ends on the same side and the 5 ends on the same side. They can't be stacked on top of each other. They have to be on opposite ends. I need to have the 3 over here and the 5 over here, which means this is the correct orientation because this is carbon number 5. So that satisfies my anti-parallel. Obviously I need a template. Let's reposition this nucleotide. Actually, let's get rid of this nucleotide because I've already got one drawn down here. So we're following base pairing rules. Green represents A, so red must represent T. Blue represents G, and we're going to use the color orange to represent C. So the next thing that I need to do is I need to synthesize my new nucleic acid strand by growing it in the 5 to 3 direction. So here is carbon number 3 of this nucleotide. A dehydration reaction needs to occur. So we're going to have another nucleotide come in. And notice that this nucleotide is going to be complementary to the one across from it, to the template strand, right? So this is T on the template strand, meaning this nucleotide is going to have to be A. Now, notice that this nucleotide is coming in as a triphosphate nucleotide. This is actually deoxyribose ATP. The nucleotide is supplying energy to fuel this synthesis reaction. And so there's an enzyme, the enzyme is called polymerase, right? Hopefully that makes sense because we're creating a polymer. Enzymes end in ase, so polymerase. this polymerase is going to pop off these two phosphates. This is called pyrophosphate, the two phosphates that are released. That's going to release energy, and that energy is going to be used to drive this synthesis reaction. And so what polymerase is doing is it's covalently attaching the incoming triphosphate onto the nucleic acid strand. All right, I'm going to pause the video so that I can draw my other two nucleotides, and we'll see this process play out. All right, here I've got another D ATP. Let's try that again. So this enzyme polymerase is going to basically detect the geometry of the base pairs. So if another trinucleotide tried to get in, so this is DATP, let's pretend for a second that DCTP, this is cytosine triphosphate, tried to get in and base pair with this T. Since the geometry of the CT base pair is essentially not compatible, it's an incompatible geometry, polymerase detects that and throws out DCTP. Only when it detects proper geometry between the base pairs will it then change shape in a way that allows it to become catalytically active. It can now catalyze the reaction. So what it's going to do is it's going to pop off that pyrophosphate and use the energy to, here we have our hydroxyl sticking off of the 3' end of the nucleic acid. Well, let me draw it like this. We're going to get rid of that pyrophosphate. We're going to pull a water molecule out and join the oxygen to the phosphate group. Right, so I erased a hydrogen. There's also an OH coming out of the phosphate group. So together, that makes up H2O. And then finally, well, let me go ahead and draw. Here's my 5, 4, 3, right? This is carbon number 3 with the OH sticking out. Finally, we have deoxyribose CTP, DCTP. It's going to come in. Polymerase is going to detect that that is a compatible base pair configuration. G and C have proper geometry. And it's going to nick off the pyrophosphate. That's going to release energy. There's going to be an OH removed from the phosphate. There's going to be an H removed from the hydroxyl. That's a dehydration reaction, covalent bond. Now we have our nucleic acid. We have just synthesized a nucleic acid. It's beautiful. So this is how nucleic acid synthesis goes. It doesn't matter whether we're making DNA during S phase or reading the DNA to make RNA so that a protein can be made. Doesn't matter what type of nucleic acid we're making. We're always going to follow these 3 rules. And as long as you follow these 3 rules, this concept will become a lot less complicated. All right, that's it for this video. Thanks for watching.